a very interesting study to do as well. So what we've done were very targeted and specific analysis, but I think it would be, like you said, very, very important to be able to explore that a little bit more and try to find other markers that would be associated to the protection. I didn't show you today, but one of the things that we also saw, one other polymorphism that we studied, which was the CD14 polymorphism, which is one of the molecules that are very important for triggering the innate immune response. That seems to have a much stronger odds ratio of protection in Chagas than the ones that I showed you today. But I think it would be very important to do that. Yes?
Aside from variants in the host, is there something about the organism that responds differently to weather, for example, and warming and colder climates versus warmer climates?
So there is not a very systematic study of the different parasite strains that were isolated in different regions of the country. There are differences in strains. There was a study that was done with strains isolated in Mexico versus the southern region of South America in Argentina. And they didn't seem to find anything that was very striking between these different strains of parasites. However, not considering, the climate in the different regions, but thinking about the genetic diversity of the parasite, there were very interesting studies showing that the strains that you see in patients with cardiac disease seem to be genetically different than the strains that you see in patients with, for example, the digestive form of Chagas disease. But unfortunately, we don't have yet a systematic study on that. So that is another thing that we'll be able to do with this population, because we can now go collect samples from these patients and isolate the parasite from the blood in the different regions and see whether there is different or not. Lee?
Yeah, I was just wondering, what's the history of Chagas disease? How far back in history is there a record of Chagas disease?
So this is a very interesting thing. So it was discovered in the early 1900s by Carlos Chagas. He was actually, it's an interesting story, he was actually studying malaria. He went to the north of our state, Minas Gerais, to study malaria, and he discovered some kids with very high fever and things that were not related to malaria, and then he isolated the parasite in 1902. Now, there are mummies that were found in the Andean region in South America that date back from 10,000 years ago that had things that seemed to be related to Chagas disease and that had parasite DNA on them. So about of this, yes.
So it's a very old endemic disease.
Very, very old.
Rather interesting in terms of the genetics of the indigenous population.
Very interesting, yeah. Very interesting in many ways, and for when you think about the adaptation of the parasite to the host, given the fact that 70% of the people live for 30 or 40 years with virtually no disease, this shows an extremely high degree of coevolution that shows a lot of adaptation between the two parts.
It just may be worth saying.
You can't ask any question, Ken.
Yeah,
I'm joking.
just the problem you mentioned about working with indigenous people is a political, social problem.
Yes.
It's not that we don't have access to the patients or could not study them.
Correct.
It's because of restrictions, legal restrictions within Brazil.
Absolutely.
So people know what that meant.
So if you want to work in Brazil and if you want to find the highest level of trouble in your life with the political people, you try to work with indigenous or the natural flora and fauna of Brazil, which, everyone thinks, oh, there could be so many different drugs and chemicals that could be isolated from them. The Brazilian government is extremely difficult in terms of opening that for research. Thanks, Ken. Yes. very important question and very difficult to answer. So it's very variable. It depends a lot on the individual. You know, some people can evolve to the cardiac form within 3 or 5 years of infection. Some people leave the acute phase with a degree of cardiomyopathy that never really gets any better. Some people will never develop cardiac disease. So it's extremely, extremely variable. We can identify the disease even in the indeterminate patients. And this disease was so common in Brazil that if you take any job in the country, this is one of the exams that you have to do to be admitted to your job. You take Chagas disease serology, so we all know, you know, if we have or if we don't have. So it's easy to identify the presence of the disease, but it's very difficult to predict when one will develop cardiomyopathy or if that person will or not develop cardiomyopathy. The hope that we have is that by finding biomarkers, such as, for example, the lowering of the IL-10 expression, can that be used as a biomarker? Can you follow that to intervene? And in that particular case, the intervention would have to be a clinical intervention for avoiding cardiomyopathy. This is the hope that we have. So we have this cohort of patients in our region that we're following up now over 10 years to see whether that evolution to the cardiac disease can be actually connected to the different levels of IL-10 or even the presence of other, cell populations that we found to be important in these individuals. But I have to clarify that there is a treatment that is given to the patients in the acute phase. So once you identify the disease in the acute phase, if you treat the patient, your chances of success are extremely high. It's about 85 to 90%. The trouble is identifying the disease because the diagnostics are really poor, really bad, and the symptoms, because we are in an endemic area, people think that they just have a cold or a fever or something like that. They're not as striking as you would hope that they would be to be able to treat those patients. Now, in the chronic phase, the treatment is very controversial because the treatment is really nasty. It has many side effects. It has worsens the heart problems, kidney problems, and so forth. So you can only treat people that have no signals of cardiac disease whatsoever indeterminate. So currently in Brazil, who is treated in the chronic phase are women that might have children, that are at an age that they can have children, and that have no cardiac disease. The others are not treated anymore. Yes?
Is there evidence that people who've been infected and perhaps cleared or become chronically not expressing it, are they protected against superinfection?
Let me see if I understood. If people that cleared naturally the infection, no, there is no report of that. There is no natural elimination of the parasite. Now, one very interesting thing is that if you take a patient in the chronic phase of Chagas disease, for you to identify the parasite, for a parasitological identification of the parasite, you have to collect approximately 30 mils of blood and culture it for about 6 months to grow some parasites. It's possible to do PCR, which unfortunately for some reason it's not very optimized yet, but the parasite levels in the chronic phase, which is when you see the cardiac disease, are extremely, extremely low.
And is there any evidence of cross-protection with other trypanosomes?
No, there's no studies with regards to that. That would be an interesting thing to say. There is one very interesting aspect with regards to that in Brazil, which is the fact that in the southern region, so Chagas is endemic in all of the littoral of Brazil, all of the all the coastal region of Brazil, except for one state, which is in the southern region, is between the two. So the southern region has 3 states: Paraná, Santa Catarina, and Rio Grande do Sul. The one in the middle, Santa Catarina, does not have autochthonous cases of Chagas disease, did not have until recently. But what they do have there is a very abundant population of another trypanosome that is called Trypanosoma hangeli. Trypanosoma hangeli is very frequent in that region, and there is no cases of Trypanosoma cruzi infection there. So that is about what we have in terms of, could that be a protective parasite to Trypanosoma cruzi. That is a thought, but we don't have scientific evidence on that.
Good, thank you very much.
Thank you very much.
I think we have a break right now. There is refreshments upstairs. But you may not bring any of it down here. If you do, you will bring down security upon us. And we have already had our initial encounter with them, and I would like to avoid that. We will begin again at 4.
Yeah, okay, there's another Boris Johnson